need to know is, one, I will urge us all to ensure that our mics are muted as we continue with this presentation so that we minimize on our background noise. In addition, we also urge our members to ensure that we input the right information while registering for these webinars so that when we are uploading your points, then we are able to upload it successfully. I don't want to take long in the housekeeping information that I will wish to share with you. We'll keep on engaging as we proceed with this presentation, and therefore you will allow me to bring in the presenter. Our presenter today is Patricia N. Mabende, who is working with the Department of Laboratory Medicine, Kenyatta National Hospital, Histocompatibility and Immunogenetic Laboratory. She holds a bachelor's degree in medical lab sciences and also a master's degree in molecular medicine. And so it's quite a big privilege to have her as a presenter today, the 6th of February 2026. Allow me to bring her so that she can continue with the presentation. Hi, Madam Patricia, are you able to hear me from your side?
Yes, I am.
Thank you so much. Dear members, I'll be your moderator today, and my name is Arthur Imari. Since our presenter is ready and set for this big job, allow me to invite her so that she can proceed, and we'll keep engaging as we continue the presentation. I also urge you to share your thoughts, your questions, and your concerns under the chat section so that as we proceed, we can take care of them, and also to the questions, we'll be able to answer them after the presentation. Welcome, Madam Patricia, welcome.
Thank you so much, Arthur, for that introduction. As you heard, my name is Patricia. I have specialized in molecular medicine, but I'm a lab technologist. And I'm excited about making this presentation because we are at this day and age, we are moving away from only hearing about molecular application techniques in research labs and in other developing countries. I'm excited about this presentation because we are going to learn hands-on and in real life how we apply molecular testing here in Kenya in one of our own hospitals to give evidence and give lab reports to not only clinicians, but also to our patients and guide surgeons on what to do. My presentation is only focusing on kidney transplantation, but its application is also somehow related to bone marrow and also liver, but I will specifically talk about kidney transplantation in this presentation. I will try as much as possible to make it as simple as possible and simplify everything. In areas where I'm using jargons, try as much as possible to remind me in your chat boxes. And at the end of the presentation, if there is anyone that hasn't understood anything, just let me know, but I'll try and make my presentation as simple as possible to make it easy for understanding for those who have no background in molecular medicine. So let me just jump right into my introduction. When you talk about kidney transplantation, you can never fail to talk about HLA, because HLA is the backbone of transplant, be it liver or kidney or bone marrow. And now we are narrowing it down to kidney. HLA in full is known as human leukocyte antigen or major histocompatibility complex. So when you see people talking about HLA or MHC, just know that they're talking about the same thing. In definition, it refers to a set of genes that code for most proteins on surface of on the cells of the human body. And its major role is in distinguishing between body's own and that which is foreign. And it's useful in our body because it's used in fighting infections. And in this case, HLA also helps in fighting our bodies. For example, if I receive an organ, naturally my body will fight it. But there is advancement in technology and science that has made it possible for people to receive a foreign organ and live on with their lives comfortably. So understanding the HLA and how it plays a role in transplant is very important. Another description of HLA is that it's highly polymorphic, meaning there are different variations of it. In a human population, you find that there are thousands and thousands of alleles that are different in a particular population, and that diversity has helped our bodies to fight and respond to a wider range of pathogens. With the understanding that HLA is highly polymorphic, disease association studies have also been done. For example, in over 90% of patients who have HLA B27 have been found to suffer from a condition called ankylosing spondylitis, which is similar to arthritis, but it's known as ankylosing spondylitis. Another association study that has been tried and tested is the HIV virus. Slow progression has been seen in patients with HLA-B57 and HLA-B27. That is just an advantage to them because they possess such HLA antigens. So that is just a by the way. HLA is divided into two classes, class 1 and class 2. HLA class 1 codes for genes in the HLA-A, B, and C loci. When you talk about HLA class 1, just think about T cells in immunology. And the function of T cells in this sense, is fighting infections directly. Once the T cells recognize that this is something foreign, they attack and cause cell-mediated cytotoxicity. And T cells are actually responsible for acute kidney rejection in case pre-transplant blood work was not done well. For HLA class 2, the genes encoded for them are HLA-DP, DQ, and the DR loci. And when you talk about HLA class 2, just think about B cells in immunology, and their function is just to produce antibodies in application to HLA and transplant. Of course, other functions are generation of macrophages and dendritic cells in fighting infections, but in this case, I'm narrowing it down to transplant. Antibody-mediated responses can also occur a few weeks to months after transplant, again, if pre-transplantation blood work was not done well. So HLA class 1 and class 2 analysis is very important to patients before they receive their kidney to minimize risks as much as possible. Okay, let's continue. What we do as a lab is, step 1 is to establish patient history. We find out if they have history of sensitization to HLA antibodies. And the first assessment that we do is ask if they have received any blood transfusion before, because blood as a product is responsible in transmitting HLA antibodies to the patients. And if the potential patient, or in this case, kidney recipient, has HLA antibodies, the antibodies could be donor specific. And if they are donor specific, there are high chances of them getting organ rejection. So we are very keen on it, and we make sure that we ask history of sensitization. Our department is trying as much as possible in improving our protocols, and in future, we are planning to screen each and every potential kidney recipient in the hope of minimizing risks for transplant rejection as much as possible. So number one, patients with recent history of blood transfusion are candidates for testing for HLA antibodies. Also, women who have had children. Once you have a child, that is also something foreign. It can immunize you and make you get HLA antibodies. Married couples as well, especially for married couples who have had children before. And in this case, for example, if it's a woman who is the potential recipient from the husband and they've had children, there's a probability of getting paternal HLA antibodies to the mother. They may sensitize this patient. So we really look at all these angles, again, with the intention of minimizing risks as much as possible. ABO incompatibility is disqualified in our department, but I know there are other hospitals in other countries and the developing world that ignore ABO incompatibility, and I'm sure they also go through various lengths to make sure that still the organ is transplanted as much as possible. But in our situation, ABO incompatibility is disqualified because we are minimizing risks for incompatibility as much as possible. So that is step one. Step two is HLA typing. HLA typing is just getting to know the alleles that are present in the HLA class 1 and class 2. HLA class 1, like I mentioned before, is HLA A, B, and C. Those loci have alleles that we will get them after a typing, and then we compare between the donor and the recipient to see how related they are. So HLA typing, we will do extraction and amplification. Extraction can be done manually or through automated means, but in our case, we do it manually and it's working well for us. Amplification obviously is done through polymerase chain reaction. And then after amplifying the DNA in class 1 and 2, remember it's HLA A, B, and C for class 1, and DR, DPs, and DQs for class 2. Once we get that, the next step is hybridization. Hybridization is just a molecular technique that is used in detection, especially in sequence-specific PCR. HLA typing cannot be done through other conventional PCR techniques like the way you amplify virus, bacteria. It is sequence-specific PCR because you already know what you want to do. So the specific sequences in the HLA-A loci, B, etc. that you want to amplify, and then through hybridization, we assume, the next slide has a visual representation of hybridization to make you guys understand better. But hybridization is, imagine after amplifying your DNA and you have it in a double helix and you want to identify it because it's not seen. So you again use heat to unwind the DNA strand to make it have two strands and come with a complementary sequence that will attach to one strand, and then you come again to have a double helix, but this time this double helix has one strand that has the DNA that you have, and the other strand has the complementary sequence that you have. Then a probe is attached to it for it to be visualized, and an instrument. In this case, we have the Luminex analyzer, which is the instrument that we use to get our results from. Microarray-based technology is what we used in our hybridization technique, and it's working very well for us. So this is the visual representation of hybridization that I was talking about. Imagine the B part of this DNA helix is the DNA that we had amplified before. So we use heat to unwind this DNA helix, and then this red part is the complementary sequence that comes to attach to the DNA that you have for it to be visualized by the Luminex machine. Of course, you have to use a probe or something that will make it possible for it to emit light once the machine is reading it, and it's making it easier for us to get our results after hybridization. So I hope that's clear enough. After that, this is what we get after typing. On my very far left, those are the HLA loci that I was talking about, A and B representing class 1 and the R representing class 2. And then this third column that has AG1 and AG2, those are alleles that are present. So for example, for this patient, the HLA-A loci has alleles at 02, 30, B 1842, and DRB1 0103. Remember in my introduction when I said HLA is highly polymorphic. Polymorphism comes with these numbers because they are so different and they range from person to person. But again, you want to look at a potential donor who has alleles that are almost similar to you to make transplantation as smooth as possible. So those are typing results that are usually exhibited in a Luminex analyzer. And this is a side-by-side report that we generate ourselves after getting our results from the machine. This is a side-by-side representation. On my left, this HLA results from a potential kidney recipient. And on my right, we have HLA typing results from a potential donor. So when you look at alleles in HLA-A, there's a similarity at 30 and 30. So those are alleles that are similar. Then we give it a score out of 6 if we were matching them against 6 pairs of alleles. So for this patient, the match is at 4 out of 6. And I can tell from the history, I think these were siblings because HLA-DR1 was homozygous because there's 11 on one side, and the donor is presenting with the same alleles. As siblings, we share alleles in the sense that we receive half from one parent and half from another. So it can be a 4 out of 6, it can be a perfect match. Parents are also potential kidney donors and recipients, and vice versa, children and parents. And they also portray a half match because as a parent, you can only contribute half to your child. And as a child, you can only receive half from your parent. So I hope that is clear. This is also another side-by-side presentation from a potential donor and a recipient, and the score is at 2 out of 6. And the same way as you get excited on the streets when you see identical twins walking side by side, this is how my colleagues and I get excited when we see something like this, because it's rare, but it's possible. This is a perfect match of a 6 out of 6. So this pair is genetically identical because all the loci from the donor and the recipient are identical. Kindly note that this match, when I say a 2 out of 6, a 0 out of 6, and a 6 out of 6, does not disqualify a potential donor from donating a kidney to a potential recipient, because like I said, the HLA antigen is highly polymorphic and it exhibits in various forms and shapes and numbers. So there are so many variations. So it does not disqualify. When you see this a 2 out of 6, I'm not marking so that to make, when you see a report that has a 0 out of 6, you see this one has failed. No, it just shows that there is a variation of the HLA alleles. So that is typing for you. The next step is antibody screening and donor-specific antibody testing for the patients that I had explained earlier are as candidates for antibody screening. Just a repeat, candidates for antibody screening are couples, mothers who've had children before, and patients who have received blood transfusion before. It is best practice to screen for antibodies because if antibodies present, they may be donor specific, and if they're donor specific, there are high chances of getting kidney rejection, and we're trying to minimize risks as much as possible because kidney transplantation is a major surgery and the emotions involved. So we want to make sure that as we release our reports to our doctors and surgeons and the renal team, our results are as reliable as possible and risks are as low as possible. So antibody screening, the principle behind is very simple. Color-coded genes that have antigens are subjected to a serum that could have antibodies. And once the fluorescent labeled anti-human IgG is added, the signal intensity is measured by the Luminex analyzer as well. The same principle is applied for the donor-specific antibody testing in case antibody screening turns out to be positive. So in this next slide, this is how antibody screening results can be. It can be positive for both class 1 and class 2 as represented in the first example here, maybe patient 1. Let me call them patient 1. Patient 1 is positive for class 1 and class 2. Patient B is positive for only class 1 and the rest are negative. You can be positive for either class 1, class 2, both, or negative. If antibody screening is done and we get a negative result, well and good, we stop from there. We proceed to the next step. But if it turns out to be positive for either class 1, class 2, or both, we test further to identify the specific antibodies that are present in this patient and analyze whether the antibodies are donor specific. So the Luminex analyzer gives out results like this for antibody screening. But in case a patient turned out to be positive for class 1, and then we do DSA analysis, we get results that look like this. So remember when I told you class 1 represents loci for A and B and C. This patient is negative for A because there is no reaction here. There's no reaction, but for B and C's, we are seeing something. So let me take the very first row as an example. C0303 is the highest reaction that this person has. And when I look at HLA-B loci, B1516 is the highest reaction that this person has. This highest reaction is seen in column that has this raw value, and this raw value tests for the antibody concentration. I'm just using the simple terms possible. The higher the antibody concentration, the higher the risk in case the antibodies are donor specific. In our case, I feel like 1,000 is high enough, a cutoff of 1,000, because as you see in these results, as the antibody concentration goes down and down and down, it gets to be negative. So the higher the concentration, the higher the risk. But again, if we get these results for patients who turned out to be positive for antibody screening, and we look at these results and compare them to the donor and see that they are not relating, then we will just release and say, yes, this patient had antibodies, but they were not donor specific. So that is safe. But if they are donor specific, it's important to indicate because this person is already sensitized And when this donor proceeds to donate an organ to this patient, there are risks because the patient is already sensitized. They already have antibodies present to the antigen that is coming, and they may get a kidney rejection. So DSA results for class 1 look like that, and DSA for class 2 look like that. Remember when I said class 2, the loci are DR, DPs, and DQs, they look like this. The higher the concentration, the higher the value, the higher the concentration of antibody. Again, we also, like the way class 1 was analyzed, you look at analysis for class 2. The DRs that are done in the potential donor, you compare and see if they are present in the antibodies in the patient. And if they are present, they are donor specific, and that's a high risk. Like I said, I'm a visual person, so this is a practical example of a patient with donor-specific antibodies. This was the typing result. I said on my left is the HLA results for the patient, and on my right is the HLA results for the potential donor. I cycled HLA B07 because I want you to mark it. Don't mind about the score because, like I said, a 1 out of 6, a 0 out of 6 does not matter. We did antibody screening for this patient, and this patient turned out to be positive for class 1 but negative for class 2. When you get such a result, you don't stop from here. You have to do donor-specific antibody analysis to find out the exact antibodies in class 1 that this patient has. And when we did, we found that this patient was positive for HLA class 1, like I mentioned before. The Bs were present, but the As and Cs were not present. Look at B07. I told you to mark that allele. This patient has antibodies for B07. And just look at the concentration. It's 23,000. Remember, 1,000 is the cutoff, but this person has antibodies, an antibody concentration of 23,000. So when you release a report like this, you let the transplant team know that this patient was positive for class 1, and the donor-specific antibodies were unfortunately detected, and the minimum fluorescent MFIs were high at 23,000, so that they may know that this is a risky patient. If they decide to go ahead with transplant, then the risk is already identified. But in this case, it's not advisable because it's very risky. This person has a very high MFI. They're already sensitized, and the chances of them rejecting the kidney are very, very high. So I'm done with DSAs. Step 4. Step 4, we do complement-dependent cytotoxicity cross-match. Just like us as lab technologists, we do cross-match in blood transfusion where we subject patient serum to potential donor cells. In this case, we do that, but we work with T and B cells. Remember T cells, when I was making my introduction, T cells, class 1, B cells, class 2. So we want to practically subject this patient with the donor B and T cells and see how the cells will behave. So the process involves manually isolating donor B and T cells and incubating them for some time. And if the recipient serum contains antibodies that recognize and bind the donor lymphocytes, cell death will occur because the cascade will be activated. So once we do that, we use vital stains like eosin. Trypan blue can also be used. I want to turn your attention to the very last box, the dotted black image that shows cell death. So vital stains, when cells die, they absorb the dye. but when they are alive, the dye will only be shown on the background, like in the first box. So the first box has viable cells, and the very last box has dead cells. So this is an example with eosin dye. This is also an example with trypan blue dye. Like I mentioned before, dead cells pick up the dye. So the representation in part A is showing live cells and B is showing dead cells. So that is also done, and the report written as either positive or negative. So before I conclude, I want to just summarize my presentation for those who joined in maybe after I had done my introduction. Our main goal is to minimize risks as much as possible in transplant biology. And what we do is look at HLA class 1 and class 2, and also harvest T and B cells to see if there is any reaction there. We do HLA typing to compare the HLA alleles in both the donor and the recipient as well. And also a very important thing that we do is test for antibodies, because if a patient is already sensitized with antibodies that may attack a potential organ, there may be rejection. So we try as much as possible to minimize risks, to make sure that transplant is done as successful as possible. So to conclude my presentation, I want to finish by saying that molecular testing has played a very important role nowadays, especially in the field of histocompatibility, in the immunogenetics. It is evidence-based. Science does not lie. And once we give our results, we are confident with the results that we give. And we know that once we do our own validations and work as a team, as colleagues, we release quality results that are reliable to our patients and our surgeons and the renal team at large. Before I finish my presentation, I want to conclude with a quote from ASHI. ASHI stands for the American Society for Histocompatibility and Immunogenetics. And it says, in transplant science, every vial, every test, every sample is a step towards giving someone a second chance at life. And we hold this thing very seriously because we usually work as a team and our procedures are very complex. So there's a lot of brainstorming, there's a lot of discussions, there's a lot of teamwork, not only with us technologists, but the clinical pathologists that we work with in our department, just to make sure that once the results leave our door, we know that we can stand by these results because we are releasing very critical information to our patients and also to our stakeholders like the doctors and the surgeons. So I hope I have given you some information. And at this point, I think I can welcome questions because I've come to the end of my presentation. So thank you. Oh, and before questions come in, I want to proudly say that initially, potential kidney recipients used to suffer a lot because there were no resources in Kenya. They used to ship samples to India and other countries and wait for weeks and months to get results. But now that is no longer a problem because, like us, our TAT is just 24 hours to get all these results. Just takes 24 hours. So I'm proud to be associated with that team that is making a difference. So thank you so much for listening to me. And in areas where somebody hasn't understood anything or something, I welcome the floor for questions.
Thank you so much, Madam Patricia. This was wonderful. We also thank our participants who have greeted us on the chat area. We have Helen Wanyama, we have Abraham, we have Leonida Akaka, we have Elvis Lukale, we have Bill Tari, we have Robert Karaoke. We also have Professor Tom Were, and he's made a comment here. Let me read. Great presentation, eye-opener on the utility of molecular technology in tissue typing in our country. Kudos. And Professor Tom Were is also requesting you, Patricia, to share your slides with him.
No problem.
We have Mwendwa Matuku. She says loud and clear, or he says loud and clear, Frederick Mwendwa Matuku from Kitui County. And we have Bahati Orembe. He says, great presentation, Daktari. The presentation can't be any better. You have nailed it with great pictorial. Indeed, beauty and brains at work. Good work, Dr. Patricia. We also have Evans Orua, great presentation. We have Abraham Lorongoi, nice presentation, kudos. We have Onyango Tieno, nice presentation indeed, great. From Frida Kari. We also have Humphrey War, she's requesting for the slides. We have Alex Makoha, brave to the point. Lilian Atieno, great presentation. Helen Wanyama, well-detailed and great presentation. We have Jared Makoha Odongo, straight to the point, I like it. We have Rosbella, she says, nice one. We have Charles Yator. She says, you have mentioned on harvesting briefly, how is B and T cells harvested? So we have Charles Yator, who's giving us our first question, and he says, he's appreciating that you've mentioned about harvesting, and so he wants to know how B and T cells are harvested. Kindly.
Oh, that's not a problem. I want to take you back to college when we were doing white cell count with a Newber chamber. We used to get whole blood, and then we put tax and Toyson's fluid. One gets you white blood cells, and the other one only gets you red blood cells. So there are reagents that are available that we use that are commercially available. So once we get whole blood, we spin and get the Buffycot, because the Buffycot is concentrated with white cells. And then we use the T and B lyse. It lyses. When you put B, it lyses T and you only get the B cells. And then when you want T, you do the reverse. So they're just commercially available reagents that you use. Then once you isolate your cells, you suspend them in media so that they do not die. And then you do your cross match with them. So I hope that is clear.
Thank you. We have great comments from Sheila Chepkosgei, Helen Wanyama, Titus Kitonyo, Bernard Rono, Wilberforce Sigei, Mesha Kinywa Ndege, Lilian Atieno. She says you are doing a good job at KNH Labs. We have Nthea Mukenyi from Kwale. We have Erastus Mwanyiro, he's requesting for the slides. Dennis Moguku says he's learned something. We have Christopher Sitati, great presentation. We have Nilesh Bagadia, thank you for the great presentation. We have Elizabeth Fwamba, wow, I've really gained a lot today. We have Albert Kamole, we have Deborah Mogi, we have Boniface Ongulu, and Boniface Angulu, and Boniface Angulu has got a question, and the question goes, hello, do you think other antigen antibodies like anti-K issue
Mm-hmm.
do affect organ transplant, and what happens in our space where we only do ABO apart from the T and B cells?
Okay, those are two different areas. Blood transfusion, when you learn blood transfusion, is when you interact with the KELs, the kids, the Duffys. But that is totally different when we talk about transplant. When you talk about transplant, is HLA and HLA alone, because HLA is what is responsible in organ transplant. And it's not only in kidney, they also look at HLA antibodies in bone marrow, in liver, in all these organs. I've also prepared slides that I may present in future about bone marrow. I think maybe you'll understand, but do not bring KELs and kids and Duffys to organ transplant. Those are entirely two different areas. And kills and Duffys do not affect. HLA does affect organ rejection.
Thank you so much. We have Ruth Ngami, detailed and well presented work. Kudos. We have Agle Ernest Danda, and he says, nice presentation. These are my questions. Question 1, how cost effective is molecular testing compared to conventional approaches? Allow me read the 3 questions, then you tackle all of them at once. Question 1 is, how cost effective is molecular testing compared to conventional approaches? Question 2, is AI or bioinformatics playing a role in interpreting transplant molecular data? That is question 2. Then question 3, how long does it typically take to get actionable results before a transplant? Back to you.
Okay, I'll start with the very last question. How long does it take for us to get an actionable results for an actionable transplant, you said? My answer is 24 hours. Like I said, our lab boasts about getting results as soon as possible for the transplant team to act on them as fast as possible. So our turnaround time is 24 hours. So once the transplant team is ready and all the stakeholders are in place, I think the transplant can go on. The second question was about application of AI and bioinformatics in relaying results. Of course, the Luminex machine is a very powerful equipment. And when we run our results with it, the alleles that we get are compared to alleles that are available in a software. And this software has all the possible alleles that are present in the population in the world. So I think that is an application of bioinformatics, because the alleles that you get from analysis are able to be compared to what is already there for them to be categorized as belonging to class 1 or class 2, and so on and so forth. So you cannot fail to apply bioinformatics in molecular biology. I think it's applied most of the time. AI, I think, is just useful in making your work easier, especially in fully automated systems. There are equipments nowadays that once you load the sample, they will extract DNA and do the master mix for you, and your work is easier. So AI just makes work easier, but you cannot fail to apply bioinformatics in molecular medicine. So you have to really understand the subject of bioinformatics to make it easier for you to interpret your results. The advantage of modern equipments nowadays is that they simplify things for you, so that if you do not understand bioinformatics well, they somehow give you results that have already chewed for you, so it's easy to interpret. The first question was about cost effectiveness. I can say about cost effective, but I don't want to talk about costs really. But I think SHA is working, so our patients are benefiting from SHA. So I hope that answers your question.
Thank you for the responses. We have Wilberforce Sigay, and here's a question. The question is, can molecular application be used in paternity testing? Back
Oh,
to you.
yes. Oh, yes. Paternity testing now. There's a subject in molecular biology called restriction fragment length polymorphism. That's an entirely different subject. There are some specific sequences that you get from maybe a parent that is supposed to be transferred to a child. And once you do amplification of these sequences, it may be a suspect child and a potential parent that you think is a parent. You just compare those sequences. And if you fail to get the same sequence from a parent and a child, you just disqualify that paternity. So yes, paternity testing is very much used, and its application is used in giving very accurate data to dispute paternity.
Yeah, thank you. Thank you so much. A good comment from Jason Mundu, Mwendwa Matuku, and we have a question from Ibrahim Bill Tari. And the question is, for kidney transplant, does it require psychological screen for donor? Maybe
yes.
you can understand that question better.
Oh, yes.
And maybe you can expand on it.
Oh, yes. Before somebody gets a kidney and before somebody gets to donate a kidney, it's a whole journey. It's a whole journey that counselors are involved. There are also other tests that are involved between both the donor and the kidney, just to the donor and the recipient, to make sure that the donor is as healthy as possible, and to also make sure that the recipient only has end-stage kidney failure and not any other complication. For example, if a potential recipient gets to suffer from TB or gets another infection, that infection has to be addressed first before now they talk about transplant. So it's a whole journey. They have to look at other organs, whether they are functioning properly, especially in both the donor and the recipient, like the lungs, the liver. It's a whole journey, so that by the time they come to our lab, it's the very last stage before they head to the transplant team for them to be scheduled for surgery. So yes, it involves a lot of stakeholders before an organ is donated.
Thank you. We also have another question from Silas Nusi, and the question goes, how do we differentiate between pathogenic donor-specific B cells and harmless memory B cells in post-transplant monitoring? And secondly, how do we quantify the relative contribution of B versus T cells in mixed rejection cases using molecular techniques?
Okay. Can you please repeat the first question? The first question was
Yes.
about.
Let me repeat the first question. The first question goes, how do we differentiate between pathogenic donor-specific B cells and harmless memory B cells in post-transplant monitoring?
In post-transplant monitoring, you in my thinking, I think you look at patients who are not doing very well. But if they are picking well, I think other tests that are followed up are just kidney function to see whether the new kidney is now working. But if they are not doing very well, I think you can go back to the drawing board. And I think in this case, you're just doing investigations to see what happened and how it can be done. For us, we do not analyze B and T cells to see the pathologic and the whatever you described post-transplant, but maybe other advanced labs do, I'm not sure. So as follow-up tests, we don't analyze B and T cells. Because remember in our cross-match, we look at the donor mostly, the donor B and T cells, and see how they behave when they're subjected to patient serum. So just like the way in blood transfusion, you use red cells and patient serum to see whether there's a reaction, the same cross match we do here, but we use B and T cells. But to go further and use molecular analysis to see whether the T and B cells are behaving post-transplant, not really. Though theoretically, if you maybe harvest maybe patient T and B cells post-transplant and look at how they are behaving at molecular level, you can get something. But for us, we are not there yet. What was the second question?
The second question was, the second question is, how do we quantify the relative contribution of B versus T cells in mixed rejection cases using molecular technique?
Okay, I think mixed rejection cases are applied in bone marrow. I'm saying that because I have slides for bone marrow where there's mixed rejection, where the chimera is not full, because in bone marrow, the recipient, you wipe out all the cells from a recipient, and then you bring in new cells from a potential donor. So once the bone marrow is transplanted to a recipient, there can be mixed reactions, like you say, mixed rejections, and there's a way the patients are managed to do that, but I don't think that literature is in kidney transplant. But again, if it is there, the patients are usually desensitized, and the level of desensitization varies from the variations in their HLA alleles between the donor and the recipient. Remember when I was showing you a score of 6 out of 6, a 1 out of 6, a 0 out of 6. The perfect score of 6 out of 6, this pair may not be sensitized as much as this pair that has a 0 out of 6. So the level of desensitization varies from patient to patient. And I think that is where the mixed rejection comes in. When a patient is not doing well, the transplant team will manage them accordingly. So I hope that answers your question.
Thank you. We have another question from Robert Musumi, and the question is, which EQS scheme do you participate in, and what has the results varied with the desired output with your providers?
We do proficiency testing, and all this is done to assure quality assurance purposes, to make sure that our results are as reliable as possible. And like I mentioned before, because we work as a team to make sure that once our results leave our door, we can defend them as much as possible, because we know that we have given out the right information and the right results to our patients and our clients. We are registered with UK NECAS, and we participate actively in all the tests that we give to our patients, and we can say we are doing very well.
Yeah, thank you. Another question from Anonymous. She wants you to clarify the process of candidacy
Mm-hmm.
and the risks involved and the logistic involved, the financial and the psychosocial requirements.
Those are many aspects that have been mentioned. When it comes to financial aspects, of course, there are costs involved in kidney transplantation. And like I mentioned before, our government has tried as much as possible to incorporate SHA, that is assisting our patients to some extent. The psychosocial support is there. Like I said before, a lot of stakeholders are involved between families and the potential donor and the recipient. There's a lot of counseling and psychological counseling that is done between the families and the potential donor and recipient to make sure that they understand what they are getting themselves into and the outcome of the transplant, how it will be, the risks involved, everything. So by the time they leave to get to the theater, they are informed with everything that they need to know. What was the first question? I've forgotten the first question.
The first question was, you clarify the criteria or the process used
Oh,
to
oh, yeah.
identify a candidate.
Oh, I can. Oh, okay.
Yes.
For our country, mostly, most people rely on relative donors, a brother, a sister, a cousin, mostly relative donors. We are not yet at a situation where there are anonymous donors, like in advanced countries where people sign off when they are alive, that when they die, their organs may be used to maybe save other people's lives. We are not yet there, but our system and as a country so far only relies on relative donors.
Thank you. Another question from Anonymous. We've seen quite a number of syndicates on illegal organ harvesting.
Mm-hmm.
And so as lab medicine practitioner,
Mm-hmm.
what type of strategies can you put in place to ensure we are able to curb such kind of malpractices in medical and in health?
I think when it comes to medicine and ethics need to go hand in hand. And when it comes to those malicious practices that you mentioned before, it's systematic. It is not a one-man show. You'll find that it's a chain reaction that starts from the head all the way trickling down to the lab and to everybody else. So the best thing is just not to involve yourself in corruption. For us, our system is working, and I don't think we've encountered any cases where um, smuggling or anything bad is involved. So, in my opinion, I think if you do not want to get yourself in trouble, just don't involve yourself in any way when organs are being trafficked and you're helping in one way or another. It's corruption and it's also ethical. So, that's just my opinion.
Yeah, thank you. Thank you. I'm trying to look at the Q&A section and the chat area, but I'm not able to see or to get any other questions that are coming. So kindly elaborate on this, the quote that you've done on the ASHI. In transplant science, every vial, every test, and every sample is a step towards giving someone a second chance at life. Maybe you can elaborate in response to the role of lab medicine in transplant, in organ transplant.
Oh, okay, no problem. Like you said before, in our lab we work as a team, and once we work as a team, we treat each and every patient and sample that comes to our lab with the integrity it deserves. And we make sure that once we get results, the results are as reliable as possible. And once we get them to our doctors and surgeons and the transplant team, we just hope that we have minimized risks as much as possible to make transplant successful, because once transplant is successful, you have given this person a second chance at life, because our clients are at end-stage renal disease, people whose kidneys have failed and hemodialysis is no longer sustainable. So once transplant is done and done successfully, because we have contributed towards that success, we are happy that we have given somebody a second chance at life.
Thank you so much. I think we have no more. Oh yes, we've okay. Thank you, Caleb. We have Caleb Achola, who has shared a comment and a question here, and allow me read it. Here we go. Have you ever come across a case of serious rejection to an extent the donated kidney have to be removed? If yes, can the same organ be transplanted again?
No, not really. Once an organ has been transplanted to a patient, it cannot be removed to be transplanted to another patient. We have not come across such a situation because the systems that we have put in place make sure that we assess those risks as much as possible from start to finish. All the various aspects all the various loopholes that are present to contribute to a rejection, we address them. Like number one, I said ABO incompatibility is a disqualification in our setup, but some hospitals in other developed countries do not see it as a problem. So we disqualify ABO incompatibility, we disqualify patients who have donor-specific antibodies, we disqualify maybe a cross-match that turned out to be positive. All these aspects are done to make sure that once our results are released to our doctors and our surgeons, the patient is safe to receive their organ unless something happened that is beyond the lab that made the kidney not be successful. But we try as much as possible to minimize risks and help this patient to get a successful transplant. So I hope I have answered this question.
Very well, very well. Thank you, Caleb, for bringing that question, and also thank you, Patricia, for the responses. I'm trying to look at our chat box, and we don't have any additional questions. Okay. So you'll allow me, Daktari, to bring you again so that you give us your parting shot before I adjourn or conclude this presentation. Welcome again.
Thank you so much for listening to me, and I thank you so much for your questions, because I feel like you listened to me and to some extent understood what I presented. I am very passionate about molecular medicine because that was my area of expertise. And we are moving away from manual things and lab results that do not make sense, because nowadays we apply technology and modern techniques, all geared towards making sure that patients are well taken care of when they get quality results. So there's a lot in the world of molecular medicine that is being used nowadays, not only in diagnosis, it's also used in treatment, in what we call, there's a word I'm looking for that I'm not getting, when you design a particular therapy for a particular patient. That is because of molecular medicine. And I think the sky is basically the limit. And I'm glad that this country, Kenya, is catching up with technology to make sure that our patients get nothing but the best. So I think that is my parting shot, and I hope to come back again to share my knowledge and expertise in this field in my future presentations, in case I get the chance to.
Thank you. On behalf of Lab Medicine Society of Kenya, we are so grateful that you took your time, made the slides, and you were able to come and present to us, give us knowledge on matters transplant science. And so we believe that we'll still need much from you, because these are areas that are still in demand in terms of practice and also in terms of knowledge. So we will always be happy and willing to give you other presentations once you are ready. Just come. So we are so grateful, and we want you to ensure that you share with the MLOs of this country such knowledge. Otherwise, to our presenters, we are also very grateful because you took your time, spared your time, and managed to attend this presentation today. I believe you've gained some knowledge that is going to help you towards advising and also giving service at your workplaces. Otherwise, we also have other presentations lined up, and we urge you to at least plan and schedule to attend our webinars as we proceed. For this particular presentation, your CPDs will be uploaded into your KMLTTB portal by in the next 24 to 48 hours. And therefore, we urge you to just be patient and wait for the points and the certificate as well. We also urge you to ensure that your emails are having space, so that whenever we send the certificates, then you are able to receive them. If by chance you're not able to see your certificates in your inbox area, you can also go ahead and check your spam. Apart from that, we also urge our participants and MLOs of this country to join our WhatsApp groups, join our Facebook page, and also join our WhatsApp channel, the Lab Medicine Society of Kenya, and other platforms that we are using to share knowledge, so that we keep learning together. Otherwise, on behalf of the Lab Medicine Society of Kenya, my name is Arthur Imeri, and I was your moderator. And I also wish you a good evening as we plan to meet you again in our next presentation. Thank you so much, and thank you, our presenter, Madam Patricia. See you and have a good evening.
Thank you.